Hello, everyone, and welcome to today's webinar. I'm Deanna McNeil, Associate Science Editor for The Scientist, and I'll be moderating our discussion. Today, our speaker, Dr. Brian Haldeman, will introduce Stunner, the only tool that combines high throughput and low volume analysis of protein quantification and sizing, helping researchers confirm concentrations, detect aggregation, and analyze multi-component samples with greater speed and efficiency. Before we begin, I'd like to thank Unchained Labs for sponsoring today's webinar. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point, and our speaker will address these during the Q&A session following the presentation. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. Our sponsor has provided us with some helpful resources related to today's webinar, and we've posted these in our handout section on the right side of your screen. You can access and download these documents at any time during the webinar. And if you've minimized the Q&A panel, simply click on the blue chat button in the bottom right corner to bring it back. This webinar will be archived on the Scientist website, and we'll send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. And with that, let me introduce our speaker. Brian Haldeman is a product manager for Lunatic and Stunner at Unchained Labs. Prior to his role, he was a field application scientist supporting the biophysical characterization of proteins and viral vectors. He received his PhD in biochemistry from the University of Nevada, Reno, where he studied the mechanics of muscle contraction. His research utilized single-molecule microscopy, pre-steady state kinetics, protein affinity and binding characterization, and protein isolation techniques. Let's make sure that your slides are up and running. Excellent. Take it away.
Great. Thank you for the introduction. Today I'm excited to talk to you about how you can hit go for fast, low-volume protein characterization on Stunner. But first, I want to talk about cooking and eating meals while in the backcountry. After a long day of hiking, it's nice to sit and enjoy a nice hot meal. Traditional methods have you cooking each ingredient in the meal separately, taking excess time and burning through precious resources like fuel and purified water. Cooking everything together is both faster and uses fewer resources. Not to mention the prep meals have gotten a ton better over the years. You could take the same approach with protein characterization in the lab. When you have a sample to characterize, it's important to know two critical questions. What's the concentration, and is my sample aggregated? Old school methods require you to measure your protein concentration in one experiment using UV-Vis, then measure size and look for aggregation in another experiment using dynamic light scattering, or DLS. This costs you the precious resources of both time and sample. Methods that combine protein characterization into a single experiment and require less sample are essential in modern labs. Today, I'm going to introduce you to Stunner. Stunner is the only system that pulls together two separate technologies, UV-Vis for concentration and DLS for sizing and aggregation. All of these results are collected from a single 2 microliter sample. If you're working with conjugates, Stunner can also measure drug-antibody ratio to know all the ins and outs of your ADC. Knock out antibody or conjugate concentration, DAR, size, and aggregate detection in one shot. Without skipping a beat, you'll know if your protein sample is good to go. All you need to do is load 2 microliters of each sample into a Stunner plate, which is a 96-well plate that has had the wells replaced with individual microfluidic circuits that you see here. Each circuit has 2 fixed path length microcuvettes to cover a wide dynamic range without the need for dilutions. If you're dealing with a full plate, you can get 96 concentration measurements in just about 10 minutes. That same sample can be read using DLS to measure the average size and distribution of sizes of particles in solution. With both DLS and UV-Vis, a full plate takes about an hour. The Stunner plates are SBS format, making them ideal for integration for liquid handlers. For even heavier workflows, hook it up to your favorite robot and have Stunner work overtime. Here we can see exactly what sample loading looks like with the help of some fluorescein and a black light. In 2 wells, we've already added 2 microliters of fluorescein. As you can see, to load another sample, you simply add in 2 microliters into that input well and the sample wicks into the plate. On the left, you can see those loaded samples, and on the right, you can see the samples after being measured with Stunner. The 2 diamond shapes are the cuvettes for 1 sample. What this also shows is, after pumping, all the sample remains in our consumable, so there's no contact between the sample and the actual pump system. No contamination is possible. Not only is Stunner super easy to use, but it also provides fantastic data. Here we are showcasing the huge dynamic range of Stunner from 0.03 to 275 OD. This means that your sample can be measured without the need for dilutions to fit the dynamic range of your assay. On the left, we see the absorbance spectra with different concentrations of purified IgG, with the telltale absorbance peak at 280 nanometers. On the right, we're plotting that absorbance at 280 nanometers against the known concentration, and show that the data is linear throughout the entire dynamic range. Once again, from 0.03 to 275 OD. Also, note that each concentration shown has multiple replicates, showing how reproducible the data from Stunner really is. Now let's dig into that dataset a little bit further to show off Stunner's accuracy and precision. We're now plotting the measured concentration from Stunner against the known concentration. Converting those values to mg per mil, this dilution series was between about 200 and 2 mg per mil. Notice that the accuracy at both the high and the low end are well within 2%. The precision between replicates was also very high, with CV values below 1% throughout the entire dilution series. Stunner can quantify sample concentrations over such a large dynamic range due to the 2 fixed path length microcuvettes incorporated into each microfluidic circuit. From just 2 microliters of sample, you can get accurate and precise concentration measurements without the need for dilutions or fluorescent reagents. Here, we really wanted to push Stunner's quantitative abilities for both concentration and size measurements. We measured 8 replicates of an IgG with both UV-Vis and DLS. On the left, we're showing the absorbance spectra of all 8 replicates, and as you can see, they overlay really well, highlighting the precision of Stunner. The IgG has an average concentration of 10.3 mg per mil, with a CV of only 0.2% between all 8 replicates. Now, I want to transition to talking about the sizing capabilities on Stunner, which are just as impressive as the concentration measurements. On the right side, we're showing an intensity distribution, which is a typical output from a DLS experiment. This shows the sizes of particles in solution and can tell us if the sample contains aggregates, or in this case, it's mostly monodispersed, well-behaved antibody, with an average diameter right around 11 nanometers. Once again, this is an overlay of 8 individual measurements in different wells. Here's a look at some example sizing data from DLS. In this case, we have measurements of 2 commercially available protein standards, thyroglobulin and ovalbumin, which were loaded at the same concentration in triplicate. Note that these proteins are different in size, which is why we see peaks at different sizes along the x-axis. These standards were run separately, and the results were overlaid onto a single plot. On the left, we're looking at the intensity distribution, which connects the size to where the intensity of scattered light is strongest. Note that the larger the 2 proteins, thyroglobulin scatters much more light than ovalbumin, which is why the peak is taller. This is a fundamental characteristic of DLS, where larger particles scatter more light. We can use that to our advantage when looking for aggregates in our samples. But usually in the world of protein characterization, people think in terms of mass concentration, usually represented as mg per mil. On the right, you can see mass distributions that normalize light scattering to size, relating the size of the proteins to a mass concentration. This confirms that our samples were in fact loaded at the same concentration. I also wanted to mention that if you're looking to max out your throughput, Stunner is compatible with robots for liquid handling and plate loading. We have an API software package available which seamlessly integrates with many types of lab automation robot systems. Now let's take Stunner and show how we can apply it to characterizing an antibody sample. Many use cases for Stunner include using it to find the right formulation buffer for each of your antibody constructs. An optimized formulation buffer will keep your antibodies in their native monodispersed form. even when the temps go down for storage or the concentration goes up for therapeutic dosing. When antibodies are in the wrong formulation buffer, this leads to more self-interaction and the formation of aggregates. Aggregates lead to issues including decreased stability of your molecule and rejection for use as a therapeutic, as they may lead to an immunogenic response after administration. There are so many factors that go into finding the perfect formulation buffer: pH, ionic strength, the right type and concentration of excipient, and the list goes on. It's also difficult to predict what the ideal formulation buffer would be without direct testing. Here we're showing a formulation screen of a monoclonal antibody at about the same concentration, about 50 mg per ml, in two different buffers. There's a commercial buffer and an acidic citrate phosphate buffer. You can see right away from the DLS intensity distributions that the protein is monodispersed and happy in the commercial buffer, but aggregates in the other one. But we can do some other quantitation on this as well. Z average is the average hydrodynamic diameter of all the particles in solution. Polydispersity index, or PDI, is related to the range of sizes of particles in solution. If there's only one size of particle, the PDI is low, and if there's lots of different sizes, then it's high. Monodispersed samples have PDIs of less than or equal to 0.1. The antibody in the commercial buffer is an example of such a monodispersed protein and is an average size of about 6 nanometers. Polydispersed samples have PDIs greater than 0.2, like the sample in the acidic citrate phosphate buffer. The antibody in the acidic buffer also has a higher Z-average diameter. The two peaks in the DLS intensity distribution of the citrate phosphate buffer shows that there are two populations of particles in the solution, one with an average size of 20 nanometers and the other with an average size of about 275 nanometers. It's possible that both of these peaks represent aggregate populations, since the one peak in the commercial buffer is about 6 nanometers in diameter. Now let's transition from the world of antibodies to the world of antibody-drug conjugates, or ADCs. Antibody-drug conjugates take advantage of targeted binding of mAbs to surface receptors unique to their target and use them as a drug delivery vehicle. For example, they're commonly used as a treatment for different types of cancer, and the antibody targets a unique surface receptor on the tumor cells. When conjugating a hydrophobic drug to an antibody, the aggregation risk goes up. It's important to track concentration, size, and aggregation of your ADC throughout development. Along with this, you also need to track DAR, or drug-antibody ratio, so that you know you are delivering a consistent therapeutic and nothing has gone haywire with a specific lot. If we're going to quantify this ADC, we can start by first measuring each component part on Stunner. The absorbance of a protein is pretty standard. In fact, if you're just measuring an unlabeled protein, this is a step you can skip. But for ADCs, it's helpful to measure a dilution series of your antibody to teach Stunner what the absorbance behavior of your specific antibody is. Separate from measuring your protein, you could take your drug and run a similar dilution series, and it's going to save that absorbance spectrum as an analyte in the Stunner software. Those spectra can then be combined as shown in the next slide. Here, we're showing how Stunner can deconvolute, or unmix, the total absorbance of an ADC, that's shown in black. Since Stunner now knows the underlying behavior of the antibody and the drug linker, it can break down that total black signal into its component parts. It will unmix them into green for the antibody and blue for the drug linker, and we can then quantify how much antibody we have, how much drug we have, and most importantly, the drug-antibody ratio, or DAR. Here we show a calculated DAR of 7.2, which is consistent with the gold standard DAR method of mass spec, which gives a DAR of 7.7. Here we are showing how you can correlate DAR with the size of your ADC in solution. We're comparing the bare mAb pre-conjugation, shown in green, along with ADCs that were conjugated at two different drug-antibody ratios, 10:1 shown in blue and 40:1 in yellow. The biggest difference you can see in the mass distribution is the width of the peak that increases with higher ratios of drug present in the conjugation reaction. One important note here is the samples are at different antibody concentrations and the amplitudes reflect this. We can't put all three on the same graph unless we normalize. All the distribution shown had a single peak. Along with the broadening of the peak in the mass distribution with higher drug ratios in the reaction mix, We also directly measure the DAR of each one of these samples. Each reaction had the same antibody incubated with increasing amounts of our model drug, an NHS ester of alexaflor 350. After conjugation, samples were buffer exchanged using our automated benchtop buffer exchange system called Unagi to get rid of any unbound dye that was still in the reaction mixture. After buffer exchange and normalization, the samples were run on Stunner where DAR, size, and PDI were all measured to find the optimal reaction conditions. To no surprise here, the higher amount of drug in the reaction mixture, the higher the reported DAR. Notice that each sample had replicate measurements and the tiny error bars showed the precision of the DAR measurements on Stunner. Along with DAR, we also measured and compared size and PDI for each of the reaction conditions and compared these with the bare MAB. The blue bars represent the size, shown on the left y-axis, and we show that the z-average size increases with higher DAR. The PDI is shown by the gray circles, and the values are shown on the right y-axis. PDI also increases with higher DAR, but most conditions besides 40:1 have a PDI lower than 0.1, indicating they are mostly monodisperse. With both of these metrics taken together, we can conclude that the higher DAR affects the stability of the ADC. With Stunner's ability to measure several parameters and its low-volume, high-throughput workflow, it really is the ultimate tool for ADC characterization and reaction condition screens. The ADC we used in this work was made of a commercially available monoclonal antibody to which we added the fluorescent molecules fluorescein or alexaflor 350. We wanted to evaluate of the two conjugates which would have the bigger impact on the stability or quality of the protein. We also found a couple of different conjugation chemistries. Fluorescein isothiocyanate, or FITC, and the alexaflor NHS ester both react with primary amine groups on a protein, attaching at lysine residues, while the maleimide reacts with thiol groups of free cysteine residues. We chose this range of reactions because we also wanted to evaluate the efficiency of the conjugation chemistries and if they impact the antibody's propensity to aggregate. We started by reacting our monoclonal antibody at 10 mg per ml with the indicated mole ratio of each dye in appropriate buffer and incubated for 1 hour at room temperature. Samples were then buffer exchanged into PBS with our automated benchtop buffer exchange system, Unagi, to remove free, unreacted label from each of the reaction mixtures. Results showed DAR for the different fluorescent molecules and labeling chemistries at 2 different dye-MAB molar ratios. From these results, we can conclude that the alexaflor 350 NHS ester is the most efficient labeling reagent. But as we have already shown, this is only one piece of the characterization puzzle. In the same experiment, we also read size with the blue bars and PDI with the gray dots for each of our samples from the same 2 microliters. Results show peak of interest mass mean diameter and PDI after buffer exchange into PBS. Most of our samples were well behaved and monodispersed with a size of 11 to 14 nanometers. One trend we saw with all samples measured is that the size increased with higher molar ratio of dye in the reaction mixture. Really no big surprises there. When looking at PDI, we see the first indication that one of our samples is unstable after conjugation and buffer exchange. FITC at the 10:1 molar ratio is the only condition that shows moderate polydispersity with a PDI above 0.1. Once we measured concentration, DAR, size, and PDI of all the different reaction conditions, we could find the best condition for each of our mock drug linkers. The best condition was chosen as a sample with the highest DAR, but did not significantly affect the size or the PDI after conjugation and cleanup. All of the ideal conditions had a final DAR around 1 to 2, even though the molar ratios in the reaction mix were quite different. The size and PDI of all the best conditions were similar to the unconjugated MAB, which suggests that there isn't a big effect on the stability of the ADC after conjugation and cleanup. This is a separate experiment that was done to highlight the sensitivity of aggregate detection with DLS on Stunner. In this experiment, we wanted to see whether an ADC was stable over longer periods of time after conjugation. To test this, we measured the size of one reaction condition directly after conjugation and cleanup, then again after 7 days with storage at 4C to see if we could detect aggregation in the sample. Starting with the day 1 data, both the intensity distribution in green and the mass distribution in blue show a single, tight peak, indicating a monodispersed, happy sample with a z-average size around 11 nanometers. This is right in the sweet spot for a well-behaved ADC. After storage for 7 days, the story begins to change, especially in the intensity distribution. Remember that larger particles scatter more light, so a relatively small population of aggregates will show up in the intensity distribution. Now this is exactly what we see in the results after 7 days, an aggregate population that is clearly present with a size around 1 micron. When looking at the mass distribution after 7 days, we can conclude that the aggregate population seen in the intensity distribution is relatively small, as we cannot detect it in the mass-normalized mass distribution. 1 week of storage in the fridge resulted in a detectable aggregation of a relatively small amount of our sample. This really solidifies that Stunner is an excellent tool for sensitive aggregate detection. One thing I have not mentioned yet is the time to get all these results on our ADC samples. Here is a screenshot of the Overview tab in our Stunner analysis software. 16 ADCs can be fully characterized with concentration, DAR, sizing, and aggregation in just about 10 minutes with 2 microliters of each sample. Compare this with traditional methods of getting all these metrics, and it's clear to see that Stunner is a game changer with respect to both time to results and sample consumption. To summarize ADC characterization on Stunner, DAR, concentration, size, and aggregation are all measured in the same experiment from just 2 microliters of sample. Not only is Stunner efficient, but it's incredibly accurate and precise. Take this data summary showing an ADC that was measured in quadruplicate. The ADC spectrum was effectively unmixed into its individual components with an error on the fit of less than 1%. This was used to calculate a DAR of 7.3 with a CV of only 0.6%. The ADC concentration was also measured from those unmixed spectra, but here we're showing the overlaid replicates of the ADC spectra to show how well they overlay. The final concentration was just about 2 mg per mil with a CV of only 0.7% between quadruplicate measurements. Finally, the size was also measured as shown in the overlaid intensity distributions. The z-average size was measured at just about 11 nanometers with a CV of less than 1% between replicate measurements. I spoke a lot today about ADCs as our model conjugate molecule. But Stunner is fully capable of unmixing the spectra of all different types of conjugates, including antibody-oligo conjugates, or AOCs, shown here on the right. These AOCs' individual components, DNA and protein, shown in blue and green in the unmixed spectra respectively, have maximum wavelengths at 260 and 280 nanometers. Stunner's intelligent unmixed algorithms are more than capable of deconvoluting the spectra, even when the peaks are close together. To summarize everything that I showed today, I hope you leave this webinar with one clear message. Stunner is the ultimate low-volume, high-throughput tool for characterization of both proteins and conjugates. With a single 2 microliter sample, Stunner combines UV-Vis and DLS to measure concentration, size, aggregation, and DAR in a matter of minutes on up to 96 samples on one Stunner plate. And of course, with all the time and resources you're saving while characterizing proteins in the lab using Stunner, you can take the time to enjoy all of our resources in the outdoors on your next backpacking adventure. I appreciate you all joining today, and I'm happy to address any questions that you may have. Thanks.
Excellent. Thank you so much, Brian, for that great presentation. We have some audience questions, so let's jump into our Q&A.
Sure.
What is the maximum concentration sensitivity of Stunner?
So I'm assuming from those words they're talking about what's the lowest limit of detection we can do. So for UV-Vis, that's going to depend on how many aromatic amino acids you have within your protein. But typically, for a standard mAb like we talked about today, typically around 0.02 mg per mil.
Perfect. Can I use Stunner to measure the size of biomolecules other than proteins?
Yeah, absolutely. So we have apps for everything from nanoparticles to viral vectors. You do just DNA, lots of different options. If you look at the resources page on our website, you can see lots of different app notes for those different particles as well.
Great. You mentioned conjugate quantification on Stunner. What types of conjugates can Stunner measure?
Sure. Yeah. Today we talked a lot about antibody-drug conjugates or ADCs, but we could certainly work with antibody-oligo conjugates as well. Those are the two most common that we see.
Excellent. And can Stunner be used in a QC environment?
Yes, it can. Yeah, so Stunner can be used in a QC environment. We have a specialized software that we can utilize here. It's for 21 CFR compliance. So you can do everything from data tracking, everything like that can be done with the 21 CFR software.
I see. Thank you. If I don't use an entire Stunner plate in a run, can I use the rest of the wells at a later time?
Yes, absolutely. Yeah, so it's pretty common only to use, a few of the wells for a single experiment. My best practices that I do is I try to protect that plate from getting any sort of contamination from dust, anything like that when you're not using it. So after I use a couple of wells, I'll mark off which ones I use with Sharpie, and then I'll put the plate inside of like a Ziploc bag, something like that to protect it from dust.
Excellent. Um, and I think we have time for one more. What is the lowest protein concentration that Stunner can measure for DLS?
Okay, yeah. So we talked about UV vis before. With DLS, once again, that's going to matter. Depend on how your particular protein scattered light. Typically, what we see is right around 0.1 mg per mil, but that's going to depend, like I said, the size of the protein and also the conditions that it's in.
Gotcha. Thank you. Perfect. Thank you so much for your time today, Brian, for that great discussion and that wonderful presentation.
Of course, thank you.
If anyone has further questions, please do consider reaching out to Brian directly. His contact is shown on the screen. As a reminder, the webinar will be archived on the Scientist website, and you'll receive an email notifying you when the on-demand webinar is available. Please also make sure to follow the Scientist on social media for updates on future webinars. I'd like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the Scientist, I'd also like to thank our speaker once more, Dr. Brian Haldeman, as well as our sponsor, Unchained Labs. Thanks so much, everyone, and goodbye.